are going to be carbohydrates. So remember, we have the villi and the intestinal glands. The intestinal glands are a source of cells, kind of like an escalator, that rise up the glands and become bound on the intestinal villi. Our food is not going down into the intestinal glands, so where it comes in contact with the enzymes is on the cells, the simple columnar cells that are covering the villi. Right? Now, intestinal epithelial cells are our source for those final stage enzymes. When we talk about sucrase and lactase and maltase, those are the enzymes that are secreted by the small intestine, synthesized. But rather than secreted into the lumen, they remain bound in the plasma membrane. So the food is going to come in contact, the disaccharides are going to come in contact with that enzyme right there at the plasma membrane of the cell, undergo their final stage hydrolysis to glucose and maltose, or glucose and galactose, or glucose and fructose, and be right there at the membrane ready to be absorbed in the plasma membrane. So they're not mixing with the final stage enzymes in the lumen. That's mixing with the intermediate stage enzymes that have come from the pancreatic duct. Okay? So on this image here, we're looking at the intestinal villus, a simple columnar cell. This is the lumen, all right? And so at the lumen, we have our enzyme. Does this look familiar to you? Remember the proximal convoluted tubule cell, where we had mitochondria and sodium, potassium active transports in the basolateral membrane, creating a sink of lower sodium inside the cell, and that drove secondary active transport at the apical membrane of sodium from the filtrate to our peritubular capillary blood. Very similar process. So as we form our glucose and galactose and fructose through a secondary active transport, a symport molecule with sodium, they're going to be drawn into the cell, and then facilitated diffusion, they enter the capillary within the villus. So that process you're already familiar with via our proximal convoluted tubule pathway. Notice the lacteal over there on the far right. That is for the purpose of taking up our what? Lipids. Okay? So simple sugars and amino acids are going to utilize this pathway just like they did in the proximal convoluted tubule. Our lipids we'll look at as a slightly different component. Looks scary. So let's break it down and look at it slowly. What do you think you're looking at here? What are these structures? Not the villi.
Oh, the microvilli?
The microvilli.
Oh, microvilli.
Okay?
Oh,
So we're going to look at the process by which we break down lactose and maltose. Okay? Those sugars, that's going to be done by pancreatic amylase. What's happened to our salivary amylase? The acidity of the stomach took care of that. All right. So in the lumen of the small intestine, we have pancreatic enzyme, pancreatic amylase. So this does end in an ASC, so it should be relatively easy to remember that this is an enzyme. Its source is going to be the pancreas, so its origin is going to be the pancreas. The site of action, small intestine, okay? And the lumen of the small intestine, so that's what you're seeing up here. All right? So we're going to have polysaccharides. This is the same thing I wrote on the board earlier. Reaction time is decreased via pancreatic amylase. And then our products are going to be sucrose and maltose and lactose. And so now we get to the site of action that we see on the board. So these are microvilli of the simple columnar cells, not the goblet cells, but the other simple columnar cells that are forming our mucosal epithelium. And the enzymes are found in the plasma membrane. That's what I meant by not being secreted into the lumen. So these are the final step.
So the
Enzymes.
the co-transport, is that just happening right while it's going right through?
So there's the enzyme lactase. It's going to break lactose down into glucose and galactose. And our co-transport, our secondary active transport molecules are going to be located right there. You know, they're floating around in the membrane. But that's why we have, as soon as that product is created, it has the availability of moving into the cell. Now, this is a schematic diagram, all right? It doesn't mean that we only have lactose enzymes, lactase on one microvilli, and then sucrase has to be on another. This is just so we can spread out the activity. It's going to be all over the place, all right? So, sucrase, maltase, and lactase. And the enzymes, so they are going to bind to sucrose, maltose, and lactose, strain that energy bond, all right, and split it into the products that we described earlier, glucose and galactose, glucose and fructose, glucose and glucose. So then these are the sodium co-transport, that's important, that I showed you earlier. So this diagram is missing the enzyme, but this is what's happening. So as we lower the amount of sodium in the cell, we would have sodium-potassium channels down here. They're not drawn in, but same as we have with our proximal convoluted tubules. So now we're going to have sodium ions and our simple sugar doing symport secondary active transport. So that's what we have in our mechanism for bringing in these sugars, simple sugars. And then we have facilitated diffusion is the mechanism. We have to have a transport protein because the molecules are too large to actually pass, solubilize, be permeable to the plasma membrane, but facilitated diffusion to move them out into the blood. Now there are some minor differences, but that's the primary focus that I want you to know for those. So now the glucose moves into the blood, the malt, the galactose, and so on. And it's going to move into the capillaries of the lamina propria in the villi. And the capillaries are going to form venules and small veins, and then eventually what vein?
Portal vein?
Not yet, not quite yet. We haven't yet left the mesentery proper.
Oh,
The,
liver?
What's the vein carrying this away from the small intestine in the mesentery proper? I know you know it. It's just
the mesenteric vein?
superior mesenteric vein to portal vein to the sinusoids that we looked at on Tuesday. Okay? So again, because this is practically identical to what we saw in the kidney, I think you have a better graph than when we started out with this. So now let's look at proteins. slides up a few years ago before I was aware of the fact that you could group images together and repeat. So I physically copied each little circle and put it in place. It took me hours. I was so, well, I wasn't mad, but I was laughing, kind of mad at myself for not, I had to redo each of these things that I could have been done in seconds. I just didn't know I could group things together. So I have learned. So now we're looking at proteins. And so what I've diagrammed here is a polypeptide. Going back to the mouth, see if there in the corner it says mouth. So going back to the mouth, we don't have any enzymes in the mouth that work on polypeptides. So no digestion occurs in the mouth. Put a picture up there just to say that. In the stomach, we have, and I'm not drawing this on the board by the way, guys. In the stomach, we have pepsin, which is the active form of pepsinogen, remember? Hydrochloric acid does that. And so this little triangle is representing the enzyme pepsin. And remember I said our protein has a nitrogen end, a nitrogenous end, and the acid end. And pepsin works in between on those carbon-carbon bonds and breaks it up into smaller proteins, or oligopeptides. They are occasionally dipeptides. This pepsin is the same thing that happens with trypsin, and that should be chymotrypsin, not chymotrypsin. So those are pancreatic enzymes that are moved into the small intestine via the main pancreatic duct, primarily. And so they're going to do the same thing. They'll work on the large proteins plus the smaller proteins that pepsin has worked on. These are still too large to cross the plasma membrane. So we can make a few smaller ones here, but primarily now we're ready for our final stage digestion. May I erase the board? You guys have that?
Hold on.
Just a simplified version of that earlier diagram. You know how much camera space you're going to have when this class is over. Okay? So our pancreatic enzymes, trypsin and chymotrypsin. The pancreas also makes carboxypeptidase, but most of it is made by the small intestine. And so we now have oligopeptide short chains. Notice they have a nitrogenous end and a carboxyl end, an acid end. And so when we look at carboxypeptidase, if it is made by the small intestine, it is also similar to sucrase, going to be found in the plasma membrane of the microvilli. Whoops, I did it wrong. So we have carboxypeptidase and aminopeptidase. And I don't know why, I just think this is cool that we have an enzyme that works on both ends of our protein. Obviously, it makes it much faster to break it down to individual amino acids. So notice that the carboxypeptidase is working at the carboxyl end, the acid end of the protein, to release a single amino acid. Okay? Now, look where the aminopeptidase works. It works on the nitrogenous end. Okay? So we have this going on at both times, removing single amino acids, cleaving off single amino acids from each end of the protein molecule till we get down to just 2 molecules, and then we have our dipeptidase that splits it in half. So here we have the enzymes embedded in the microvilli, similar to what I drew on the board. And the individual amino acids released by the carboxy and aminopeptidases. And the carboxypeptidase is here as well as up here, because remember I said carboxypeptidase is also released by the pancreas. So it's going to be in the lumen as well. And then dipeptidase, aminopeptidase, and the intestinal carboxypeptidase are embedded in the plasma membrane. And the same secondary active transport with sodium that we saw in the proximal column of the tubule will drive the amino acids carry the amino acids into the simple columnar cell. And then they're transported into the capillary as well. Okay. So that's our protein. Same activity as I drew for you at the very beginning when we were looking at the overall picture. The key difference with these diagrams is just the location of those final step enzymes. And again, without those final step enzymes, without the enzymes of the small intestine, we wouldn't be able to transport disaccharides. We wouldn't be able to transport dipeptides. They would stay in the lumen. So we need the small intestinal enzymes, but before those work, we need the enzymes transported along the main pancreatic duct. All right, that brings us to our lipids. And let's look back at what's happening. May I erase the board? I don't think there's that much information up there, but you want it? I know you want the information. You want the picture. Picture. So the mechanical churning of the stomach is essentially what you're going to do in lab today when we look at the effect of bile salts by taking some vegetable oil and water and shaking it up in a paraffin-covered test tube. That's mechanical digestion. All right, that's physically breaking down the oil globule into small droplets. So this is our large fat globule. Okay, so if you had a cheese Danish this morning, or you ate a glob of butter, deep fried butter, or whatever. I've never tried those, but I've heard they're good. Then the mechanical churning in the stomach is going to break that down into individual smaller droplets. Now that would recoalesce into another large lipid globule in the intestines as it moves through if we didn't have the presence of the bile salts. And salts are charged. Okay, salts can dissociate to charged molecules. So that's what's happening here, is there is a charged region and a non-charged region, because obviously our lipid is not going to bind to a charged region. And so the non-charged region becomes soluble in the lipid droplet, and the charged region extends outward, and those charges repel each other. So that maintains the emulsification. The bile salt does not chemically break down the lipid droplets. It's still a lipid. Okay? All it does is maintains the increase in surface area. So if you were digesting a lipid with and without bile salts, and after 30 minutes you don't see digestion or very little digestion without the bile salts, that doesn't mean that the enzyme, the digestive enzyme, isn't present necessarily. It just means that you have relatively less surface area. Therefore, it's going to take a while for the enzymes to gain access to all of the carbon-carbon bonds that need to be broken. And so this is increasing surface area, not doing chemical digestion. So we have then these components that are covered with the bile salts, and this is what is going to enter into the lumen of the small intestine, and just the droplets, and then because of the bile from the common bile duct via the ampulla of water, the salts are associated, and so we get these micelles. Not as in my cell versus your cell, but a lipid droplet with its associated salt. So this is not a coated molecule, but a charge-coated molecule. Okay, that repels other components. So now, as we get to our columnar cell, actually I'm going to draw it sideways. I think you have these drawn sideways in your diagram as well. Let me draw that quite a little bit bigger here. A little bit longer. So our enzyme for digesting fat is pancreatic lipase. So as pancreatic lipase, and that's pancreas, not small intestine, okay? So as pancreatic lipase interacts with the surface of our micelles, it is going to form fatty acids, and it's going to form monoglycerol. So glycerol with one fatty acid. So typically from a triglyceride we have two fatty acid molecules and a monoglyceride, a glycerol with one fatty acid. And those are permeable, they're lipid, so they are permeable through the plasma membrane. And they enter into our columnar cell just by crossing through the phospholipids. So now we have 3 fatty acids and monoglycerol inside the cell. And they come back together again. So they reform triglycerides after going through that whole process. They go back to a triglyceride. And triglycerides, remember, fats are not going to be soluble in the water of our plasma or even our extracellular fluid. So for transport, they are coated with proteins. And they do that by passing into the Golgi apparatus that contains proteins synthesized via the ribosomal ER. So looks like a Golgi apparatus, doesn't it? If it did, I wouldn't have to label it. So now within the Golgi apparatus, our lipid is coated with a protein coat. And this is now called a chylomicron. So the lipid and salt association in the lumen are the micelles. The lipid and protein coat are chylomicrons. That's surrounded by the plasma membrane from the Golgi as it pinches off. Remember how Golgis pinch off secretory vesicles? And those can either go to the mitochondria or to a lysosome or to the surface of the cell. So these go to the surface of the cell. Their membranes fuse. And the chylomicron is released. But as we discussed earlier in the anatomy of the digestive tract, this is too large to pass through the simple squamous membrane of our blood capillaries. And so instead, we have the simple squamous epithelia of our lacteals which are plenty large enough for white blood cells and bacteria. So they're large enough for this structure, chylomicron. To enter a lacteal. This is why we have our lymphatic fluid from the digestive tract is rich in lipids, which does not go directly to the liver. All right? The lacteals go to the cisterna chyli, which goes up the thoracic duct. The fluid then goes up the thoracic duct into the left subclavian vein and brachiocephalic, the superior vena cava, to the heart, to the lungs, and back. Just doing a review, right? And out the aorta to the celiac trunk, to the hepatic artery, common, proper, and then it joins with the portal vein in the sinusoids. You guys know all that? Yeah. So, we have portal vein and hepatic artery. Remember, this is the one site in the body where we have venous blood and arterial blood flowing in the same direction. So the portal vein is rich in simple sugars and amino acids taken, like we saw on the two previous slides, via the capillaries. And the hepatic artery is rich in lipids and oxygen. So when we look at our models, and I told you this when we were talking about the portal vein and the hepatic artery way back in the cardiovascular system. So it is there in some distant file in your brain. So, rich, hint for a lab exam, remember our multiple choice questions. So the blood capillary is going to be rich in amino acids and simple sugars. The lacteal is going to be rich in lipids. And through both the vessels, portal vein and hepatic artery, we get those three classes of nutrients into the liver, where they mix within the liver, okay, in the sinusoids. Now obviously with the fetus, all of those are carried via the umbilical vein, okay? We're almost done. For today. The large intestine. Nobody really rids themselves out well for a colonoscopy here. It's easy little bits of fecal matter. pH of 8 to 9, that's what we've been having with our small intestinal components. We don't have the release of any digestive enzymes from intestinal cells. Obviously the bacteria have their own digestive enzymes. And so as far as our own cells are concerned, the mechanism here is absorption of water. So we are absorbing water and any of the vitamin K, primarily synthesized by the E. coli that are present here. So we don't digest the plant fibers and so on. This is micturition reflex. This is the defecation reflex. I'm not going to go over it because we've done it already for urinary system, but the slide is on there. Same type of stretch receptors. Parasympathetic is going to activate peristalsis and cause internal anal sphincter to relax. What's the nerve that's going to cause the external anal sphincter to relax? Pudendal, and primarily the inferior rectal branch of pudendal. You can see it on our models going straight towards the anus. Okay? This is just a summary of everything we talked about today. So when you go back and review today's material, we've listed, or I've described, enzymes. So as you're thinking, remember, I encourage you to try to remember as much of the lecture before you actually start studying. So you can take it through mouth to esophagus to stomach, or you can just say, okay, what are all the enzymes that I can think of? What are all the hormones that I can think of? And then, again, before you look at your lecture notes, can you take that enzyme from the list of enzymes that you come up with and place it in the proper organ, the organ it came from and the organ where it works. All right? So again, on your lecture exam, you will have a table, similar to what is on the sample exam. You will have a table where some of the information will be given to you and some won't. So I might give you the site of origin, the site of action, and its function, and ask you to tell me what the molecule is.